This video aims to describe how cloning can occur. Now, clones are individuals that are genetically identical to each other. There are different ways of producing cloned animals, but currently the most successful method uses a process called nuclear transfer. To do this, you need a donor egg. In this example, you're going to get the egg from this black-faced sheep here. You then remove the nucleus from the egg, so basically it's an empty egg shell. You then take a body cell from the sheep you want to clone. Here it's the white-faced sheep. You remove the cell. That cell is containing all the DNA. You can then remove that nucleus of that cell and put it into the empty egg cell. You would fuse that nucleus into the egg cell with electricity. Then, this is done in a petri dish. You would get that egg to divide into an embryo. You can then implant that embryo into a surrogate sheep. That surrogate sheep, after the gestation period, will eventually end up giving birth to a clone of the white-faced sheep. It's a clone of the white-faced sheep because that's where we got the DNA from. The DNA was found within the cell donated by the white-faced sheep. It's actually quite difficult to clone an animal. For example, Dolly, the first cloned mammal, took 237 attempts before she was created. And even then she grew older much more quickly than normal. Despite these problems, cloning is useful to make a genetically identical copy of an adult organism that has desirable characteristics. For example, bulls whose sperm produces high quality calves are valuable and so are worth cloning. Cloning is also used to produce copies of individuals with a genetically engineered trait, such as cows engineered to produce human insulin in their milk. Because this will guarantee that all offspring will have that engineered trait.